Thank you so much. Many people talked about ecosystems. You need a team. So I want to start with thanking some of the thought leaders behind this project, which is Lars Ottersen, Thomas Lundgaard, both are sitting there. We have Alfred Sporman, who's not here today, and Bezat Partoon. Bezat is somewhere. There he is. So thank you for them. So it is really a team effort, as you can imagine. So on the screen, what you see right now, you see several of the reactors that we built before we built the pilot plant. So these are 2-liter reactors, 2-stage systems, and I'll explain more about that. And you can kind of see it's a little bit difficult maybe, or it's a little bit complex, but not that complex, because you see the 2 reactors, and I'm going to see if I can, so these are the 2 reactors, but everything nicely fits into a chemical hood, so it's not that complicated. And here you see the 2 reactors, and then here is 1 reactor. Not that complicated. So now you're scaling it up. So when you scale it up, actually this is only a 50 times scale up. So this is going from 2-liter reactors to 100-liter. Not that much of an increase, you would think, but it makes everything much more complicated, as you can see here. Now, if you scale it up now another 10,000 times, you would see just the reactors and you see all the other stuff. You don't see them, and it looks simpler again. But you have to take these different steps in order to scale it up. So this is the plant in Viborg, paid for by Cork. Thank you so much for that. And I want to acknowledge also the people who run it, because that's the software I call. That's super important. So Alberto Robots and Monica Tomovska are running it right now, and Torben Larsen and Merit Sørensen, who are the technicians helping in that project. And tomorrow, this plant will be opened. So that's a big day. So why am I talking to you about protein? I want to spend maybe only 30 seconds on the problem. The problem is huge. Right? There's too many things to mention. One of the things is the greenhouse gas equivalents about 26% through agriculture. And a lot has to do with animal husbandry and the need for us to eat animal protein. There is a solution, and the solution is single-cell protein. And this is not new. People have worked with single-cell protein forever. We've been making cheese from filamentous fungi. We've used yeast. That's what I will talk about today through bread making, and we all love beer. So yeast is not something unfamiliar to us, and bacteria in yogurt. So we know single-cell protein. I also want to point out Lisa Schmitz here, who worked on some of the data that I'm showing you, and also Professor Michael Reichlich's lab at TUM, who helped us with measuring of vitamins. And I'm going to not only talk about proteins, but also about vitamins. Okay, so conventional versus single-cell protein. The top figure here shows that we're all using CO2. Like we're thinking like, oh, this is so great, CO2 to protein, but conventional agriculture also uses CO2. They just grow plants and they produce sugar and plant proteins and the intermediates that then the animal takes for protein for their food. We, in my specific project, we also use a 2-stage process. We take CO2, we feed that to bacteria, it produces acetate, and we had already a nice talk earlier about the company again that makes acetate. And we, in addition to just the acetate, we grow yeast for protein for food. Water is used in conventional agriculture. Sunlight is used also in conventional agriculture, but we also need sunlight because we need a lot of electric power to make hydrogen to make acetate, to make the protein. So we are energy intensive, and this can only work if we have renewable energy. I love this paper, and this is from Dorian Leger, and he's also the person later on, I'll show you some information on the techno-economic analysis he did. He was also involved in that. And I love this figure. So he decided to compare 3 different ways of making alternative protein. So all 3 are much better than animal protein. Animal protein is not even on this slide. And he compared, what if I make single-cell protein based on PV? So it's more what we do, air to protein, CO2 to protein. You can also make single-cell protein from the middle figure here. We can make it by just growing maybe sugar beets or using conventional agriculture to make sugars that you then feed to single-cell protein to make the protein. And finally, on the third one is just protein from soybeans, right? Very cheap type of protein. But you can immediately see that you need much more land for that. So if we go to the left, the PV-driven single-cell protein, which is something that we do, so you have a lot, you use the land in the sense to have PV, photovoltaic, you have DAC, the DAC, and you have the microbial cultivation. And if you now look at 1 hectare, I put 1 hectare full of PV, DAC, and my microbial cultivation, I can feed 520 people. That's what this shows. In the middle one, it shows sugar beet. And this is also, there's a product called corn. Maybe you've heard about it. So this is done at the full scale. You can feed about 90 people. But if you grow the protein by plants, just by plants, only 40 people. So you immediately see that the impact that we have is on the land use part. Conventional agriculture uses a lot of land. And what I'm talking to you about is not new. There is companies already in existence that are further away. There's something called solar foods. Sometimes they call it air food. And you can read about all these different startups and what they're doing. So most of the startups in that was described in that paper, they use a one-stage system. And I'm gonna turn around a little bit. So we have a lot of electric power, we have water, we have an electrolyzer. That's often common of power to X. We produce hydrogen and oxygen, and we have a CO2 source, could be many different sources, and we mix all of those into a gas fermenter. They all come together, so we have only one stage. And the bacteria grow in there, and then they produce the protein as part of the cells, right? So we do it a little bit different. So we wanted to use some knowledge that we have on the efficiency of the process. So if you use an anaerobic CO2 reduction, it's a much more efficient process biologically. It's called part of the Wundendal pathway for the people who know. So we have the electrolyzer still, we have hydrogen and CO2, and we only use the oxygen later on. So this process here is anaerobic, so no oxygen in there, and we produce acetate. We use an organism called Thermoenterobacter kiewii. That's a thermophile. It grows at 65 degrees Celsius, and it grows very well, and it's not genetically modified. So we chose to not go into genetic modification. This is all wild type. Then you have the second stage where the acetate is being fed plus the oxygen to Saccharomyces cerevisiae, so normal yeast, which is something that's already safe and we already know. If you look at the energetics of both processes, this is where we felt or the advantage could be gained. Then if you look and you do the stoichiometric calculations and with the thermodynamics, then theoretically we can make 9.5 milligram biomass per kilojoule of energy. While in our process, because it was more efficient, I told you, we can get 24.4 milligram biomass per kilojoule. So we can do better than the single stage. And that's why we're doing this. But of course, we have some other issues because now we have 2 stages. Maybe that's more complicated. The other thing that I want to tell you about is that we will produce in the second stage CO2, and that needs to be recycled back to the first stage. So there are some complexities, and that's exactly why we are doing the scale up. We need to figure this out and how to make this work in a scaled up version. Okay, if you look in our lab, we have then the 2 reactors, stage AAP, T. kiewii at 63 degrees, yeast at 30 degrees, and you see the pHs. We are not adding yeast extract. So if you start having very expensive medium, this will not work. So no yeast extract in the first, and no vitamins. So T. kiewii makes all the vitamins. And in stage B, we have no yeast extract, but we do have to still add some vitamins. So this is something to work on also in the pilot plant to make that more efficient. So the talk is about protein, but I'm going to talk about vitamins because we want to make food, not just protein. We want to have a nutritional food. And in our endeavors, what we found out that yeast is known to already make folate, and folate is one of the most important vitamins for humans. And they do this and it is known to do it with glucose. We have acetate, so we tried and see if we're producing these vitamins also with acetate. Now, it's an important vitamin because it's necessary to make our DNA and RNA, and humans need it all the time. So the daily recommended intake is about 300 to 400 micrograms, and you need it all the time. In our diet, where we live, I just had 2 wonderful sandwiches. Probably is already a lot of folate in there. But in the developing countries, this is not the case. So maybe we have something to work on, and the yeast could go for protein, but also for vitamins. There's many forms of folate, and I'll show you in the next figure a little bit of data from this. So if you grow T. kieferi, we actually producing a lot of, T. kieferi produces a lot of vitamins. But what we found out, it was all in the cell, and in the supernatant of the reactor, there was nothing. And also we found out that this is very hard to extract from the T. kieferi, so maybe not necessarily for our food. But luckily, what we also found is that the yeast produces quite a lot of folate with acetate under continuous conditions. So that's good news. Also, they keep it very nicely inside the cell. Nothing is in the supernatant. So we have a product that we would dry, and if we don't heat it, then the folate can be in the food. So we see that it produces a lot of folate. We also find that this one, which is the purple color here, it's the vitamin that's present in the food, that there's a lot of it. And this is the right vitamin that we need. So that was good news as well. If we now ate 6 grams of dry mass of our product, so this is the yeast cells themselves, dry, then for 6 months, this is about half a tablespoon per day, we provide the recommended intake of folate, is 400 micrograms. So we need a little bit of our product to provide this folate for us. And we get a small amount of protein from that as well. If you look at the nutritional comparison, and I didn't know that, I learned something, beef and pork, there's very little folate. So if you eat only beef or pork, you will die. There's just not enough folate in there. So don't do that. You need other sources of food. But if you eat our yeast, and this is based on 85 grams of dry material, you would actually get too much folate. I don't know what that will do to you, but that's not really what we want. So we also performed a techno-economic analysis. We did that with CX Bio and a team of about 10 people. And I've learned one thing, that it's really great to work on a techno-economic analysis in a large team, rather than do it by yourself or something like that. So we normalized everything to 1 gram of product, and that product would then, under the conditions of folate production, 43% protein, and you can see the rest of the components. We don't treat it with heat. So if you eat 85 grams of yeast to get your protein, you will have to heat treat the yeast, because otherwise you have too many nucleic acids and you get gout. If you only take 6 grams, you don't need to do that. So you can just mix this with other foods, and you get enough that to for your daily recommendation requirement of folate, and you get a little bit of protein. So that's what we did here for this TA. We didn't do the post-treatment of heat treatment, and we found that at the biomass production rate of 1.5 grams per liter per hour, in techno-economic analysis, where we scale up about 700 times of the process that we have, we can feed about 5.6 million people per year with this folate and 5% of protein. This is under a capex of about 200 million and operating expenditures of about 92 million. And a payback time on this model with vitamins would be about 5 years. And if you look at the comparison with other protein sources, we chose a market price of $20 of kilogram of our product coming out with a minimum selling price of $8.41. And if you don't compare that, the green one is 8.41 and this is the $20. You see that we're comparable to some other protein sources, but we would, if you then move to the right, we are definitely much cheaper than the folate powders or pills that you could have. So you can see in this development of pilot scale to the demonstration scale and to the real scale, this transfer, what we're trying to do is try to find a product that has a little bit more value than just the protein. To have this first step go a little bit easier. So that's what we kind of did here. And if you just make proteins, the payback time would be about 7 years. So that has a comparison. So the take-home message, this is almost my last slide. Yeast biomass can be an excellent source of for both protein and folate. I hope you believe me with that. Yeast biomass has superior folate content compared to common protein sources. Optimization still have to happen. We're doing that through a pilot plant of which you saw the glamour picture of the pilot plant, and this needs to be happening during scale up, and we can feed about 5.6 million people with yeast biomass based on the requirements of folate in this 700,000 times scaled up facility. And the final slide is just thank you to everybody, and especially in this case for CORE, the Gates Foundation, that are helping fund this work. So thank you so much for that.